very happy to present today. I hope you had a great day to date. We'll be together for the next 30 minutes. I will introduce you to MIMS. So a little bit of history on MIMS. MIMS is a 3-year-old company, software company based in Montreal. We have today 30 employees. We raised around $3 million in equity, and we signed over the past 24 months for $4 million contracts. And the mission of MIMS is really to develop what we call systems biology software to help life science companies develop a more efficient and personalized medicine and agriculture. And as you will see, we're across the board from biopharma and agriculture company. So what we're doing exactly for them, we are helping actually the life science companies doing a very critical transformation process of their R&D. The biopharma and agriculture company are moving to identify their next blockbuster drug, seed and breeds from molecular biology-driven R&D into systems biology, machine learning, and computer driving motor to do their R&D. And this involves actually more and more modeling biological systems. So this modeling involved the integration of different layers of information. You have here multi-omic data, so genomic, epigenomic, transcriptomic, proteomic, metabolomic, and so on. Information on the phenotype of the system, whether the system is a human, so a patient, for example, a cow or a plant. And also integration of the relationship with this system with its environment. And this allows the generation of a graph that represents the patient and its interaction with its environment. This involves also integration of a lot of knowledge coming from the public space and mostly from the literature. Now, in this transformation process, even biggest organizations are facing 5 major challenges. The first one being that they have difficulties to build and maintain a multidisciplinary team with expertise in bioinformatics, systems biology, and machine learning that are required to do this modeling. You guys and Mila have a lot of experience as a mathematician usually to discuss with a lot of people, and I'm pretty sure that you had the experience of talking to biologists and witness the fact that really it's not easy to understand each other. So that's what these companies and these organizations are facing right now. The second challenge is really the fact that to be able to do this modeling, the organization need to capture, maintain huge knowledge base that are integrating knowledge coming from the public space. And this knowledge is for half of it into unstructured formats, so articles and text. And for the second half, it is structured, but it's structured in databases and in hundreds of databases that are dispersed worldwide. So these organizations have problems to aggregate this data and also to maintain it and to update it regularly. The life science industry is also characterized by very heavy and sensitive data. So they are facing computing challenges, and they're also facing security challenges for very sensitive data, particularly medical records of patients. And the last but not the least, when you talk about the transformation process, you talk about adoption problems. And 80% of the employees of this organization that are involved in R&D are life scientists. They're not computer scientists, and they don't have expertise in coding. So you need this new approach to be adopted by this population of scientists. So at MIMS, we help them resolve these 5 challenges with our platform by providing them with ready-to-use solutions for biological systems modeling, integrating systems biology, bioinformatics, and machine learning. These solutions are used in the biopharma industry, as you'll see in the next slide, for population stratification, target and biomarker discovery, for example. The platform is also providing them with a comprehensive and up-to-date knowledge base that is built from structured and unstructured sources. For the unstructured sources, we use NLP. Otherwise, we capture and integrate and curate automatically and manually different databases that we integrate into a massive ontology. The third thing that this platform is bringing is cloud computing infrastructure that could be deployed, retracted, that allows the data to be geolocalized and also allows federated learning to occur so that the data are not moved around and there are some learning that can be done at every site. And the last, the human-machine interaction that simplifies very complex process, particularly the systems biology one, and allow life scientists to really dig into their results and understand what is the root of the analysis that is done, particularly in the machine learning process. And all that is possible because our software is built around a kind of AI that is a symbolic AI that we call goal-oriented massive multi-agent systems that are used for reinforcement learning, for example, but that are operated here as an organizational center and core of the software. So our platform to date allow a biopharma and agriculture company to have access to different solutions for target discovery. So the first one, target discovery. So we are bringing patient stratification very early into the process of target discovery, allowing biopharma really to identify the heterogeneity between patient population in their target discovery process, so that they are this helps them a lot to have like more to prioritize target that will be more efficient and consequently have lead that will be more efficient for a subgroup of population, and in advance to know in which portion of the population having a certain disease this lead will function on. And another site where we have a lot of traction in the biopharma industry is really before clinical trials. Right now, where we help corporations that have assets for different indication to prioritize in which to prioritize the indication they should pursue with their clinical trial. In the agriculture space, the equivalent of these solutions are really into precision breeding and precision farming, and I will show you examples of the different solutions. So how come we ended up with this solution? Actually, MIMS is really the results of the integration of the expertise of the three co-founders. I myself, a former Canada research chair in systems biology. I worked for 8 years at McGill and for 13 years at UQAM. I've been actually blessed to come in Montreal from south of France, the year where the first human genome got sequenced to do my postdoc, and totally fell in love with this systems biology that was just like coming with what we call at first functional genomics, and have as mentor Mark Vidal from Harvard that really helped me into this path. 8 years ago, I met Abdullah and Michael, actually independently. Michael is an AI specialist. His specialty is really in autonomous systems and goal-oriented massive multi-agent systems. He implemented these systems in various robotics projects in the aerospace and defense in Europe, and also incorporated this solution in an e-commerce when he arrived in Montreal 15 years or 13 years ago. Abdoulaye Baniri Diallo was actually my collaborator when I was a university professor. He's a bioinformatician. He received in 2018 the Next Einstein Fellow Prize for his career at the interface of bioinformatics and machine learning, and is extremely active in both biopharma and also agriculture field. The technology of MIMS and our platform is based on more than 30 peer-reviewed publications on the different areas that are required to develop this kind of technology. So bioinformatics, systems biology, and multi-omic data integration, machine learning for target, drug, and biomarker discovery, ontologies, cognitive informatics, natural language processing, and the goal-oriented massive multi-agent systems. So how does this work and what are we doing? So this is an example of what we're doing for population stratification, target and biomarker discovery. Basically, the pipeline goes as this. So we build API to plug our platform to the source of the data of our client. These data could be raw sequencing, mass spectrometry, text, data. They could go in a structure pipeline where they go through bioinformatic pipeline, quality control bioinformatic pipeline. Could go also through an NLP pipeline to extract and structure knowledge. Then after it goes into our systems biology engine. This systems biology engine actually allows these pieces of data to be integrated one to another, and also with our knowledge base, MIMS knowledge base, that is actually built from different components. I'll go back on that slide and just move right now on the knowledge base. So the knowledge base is basically a knowledge that is a gigantic ontology where the knowledge is organized in different layers that are the different layers that are representative of biological systems. In these layers, we integrate different databases. We have to date 41 databases that are integrated into MIMS ontology, as well as knowledge graph from directly from the literature that is built using NLP and ontologies. And also we integrate data from patients that we collect from public sources. So once this integration is done, this integration actually is done to generate two kinds of graph, what we call a genetic interaction graph, which will capture for a given system, a tumor, for example, or immune system, the functional relationship between genes. And this is inferred through supervised machine learning techniques. And we're also inferring a systemic graph that is capturing the multicellular aspect of the system and also its interaction with its environment. And in there, you have also the phenotypical data. So the clinical data are integrated in there. These two graphs are interconnected. They constitute what we call the ID graph. And after that, we use graph embedding and graph classification so we generate one ID graph per patient. And after we use graph embedding and graph classification techniques, that could be either unsupervised or supervised to classify patients based on their graph or sub parts of these graphs. And after that, we use pattern capture to identify different kind of things, whether we're looking for targets or biomarkers. There are different ways we're after manipulating these graphs. So just a focus on our knowledge base, because this is kind of an interesting assets that biopharma really appreciate and allow them to actually enrich a lot their proprietary data. So we have a process where we implement actually our knowledge base. We give access to our knowledge base to clinicians. And this will start actually from March, where clinicians will be able to serve this graph and will help us to give feedback on it and some kind of validation. And this knowledge base is then after nurture our solutions for population stratification that are used by biopharma for target and biomarker discovery. We have different case studies that show that our approach is very good to identify targets. So we have shown with a success rate of 45% in validation lab that our GI graph and ID graph, the first neighbors of disease genes here, genetic disease, genes associated to genetic diseases, or potent therapeutic targets, and we have been able to validate that in the form of Duchenne muscular dystrophy and a rare mental retardation disease. We have also shown that doing this target discovery on a graph on stratified population here in oncology by comparing a graph, for example, here on TNBC and luminal A, increased by 8 to 10 times our ability, the performance of identifying therapeutic targets here for controlling metabolic reprogramming of TNBC. So to date, this technology is used on different projects. I'll just make a very brief overview of what we're doing currently. So our technology is used for 2 clinical trials in oncology in collaboration with Mark Basic at the Jewish General Hospital. This is a project that is funded by CQDM and in collaboration with Exactis as well. This project aims to identify biomarkers, to identify subgroup of TNBC patients that are chemo resistant and that are bad or good responders to capecitabine, which is a second chemotherapy that is to date given to all patients. And the second project with Dr. Mitrakos at the MUHC, funded by MedTech, where the goal here is to identify biomarkers for good responders to angiogenic treatment on patients with colon cancer and liver metastasis. For this project, we're collecting clinical data from tumor. We're collecting genomic, transcriptomic, microRNA data, and from exosome purified from blood, we get microRNA and proteomic data. And the goal here is to primarily try to find biomarker, circulating biomarkers, so biomarker from blood, and if not possible, biomarkers from tumor resected from patient. We have also a project with a big pharma. I cannot, the name of this pharma is still confidential, so I will not be able to disclose it, but the project is a project of target discovery for autoimmune disease. In this project, we're collecting clinical data, genomic, epigenomic, transcriptomic, proteomic, metabolomic, flow cytometry data from around 2,000 patients from blood. Here, the objective here is really to model the immune system of this patient, to identify subgroup of patients with the different autoimmune diseases, and after that to identify new targets in every subpopulations. In the agriculture side, we had like a project with Navaya, which is a high-tech cannabis company based in the North Shore of Quebec. This company actually has a vision really to go into multi-omic, which is not that frequent for cannabis companies. So it was really good to be involved in this project. So in this project, we're collecting genomic, transcriptomic, metabolomic data from plants in the growth facility, and as well as data from sensors. The goal here was really to model the plant, to help them to improve their production, so optimization of plant production, and also to optimize the breed. So we are really here in precision breeding and precision farming for these clients. Another project in the farming space. So that was actually one of our first projects. We are actually starting a second project with the same group of people. It was a project that is funded by Genome Canada and recently got refunded actually by Cribic. In this project, in collaboration with UQAM, Université Laval, McGill, and Valacta, which is the organization setting the price of milk and doing a lot of farming advisory for farmers in Quebec. We are collecting from 1.6 million cows, genomic, health, production, food, and climate data. The goal here is really to optimize animal health, to optimize farm production and profitability. In the second project, we're actually using techniques to capture anomalies in dynamic data in order to identify and characterize the resilience of the cows. So that's a new project. But in the first project that we carried out with them, we've got publication, and we are actually functioning in this project. A lot of the research associated to the project is done actually in the lab of Abdoulaye, our CSO, and we're putting all that in industrialization at MIMS and incorporating it into the platform as we talk. So for this project, we generated a knowledge graph that contain elements on pedigrees, production, genetic health, herd, nutrition, and environment. We used LSTM deep learning model to be able to, from input production input that was dynamic patterns, to output the production for the next year, and actually being able to infer with 90.6% accuracy whether a cow will be productive next year. Here is the curve. Actually, it's a little bit hedgy here, but it should be like rounder. It's just that we don't have a lot of point in this part here. But this shows actually that models are becoming extremely efficient from 100,000 cows. So basically, we didn't need like the 1.6 million cows to be able to a very efficient model. And this model is actually performing a lot the ARIMA model, that is a model that is currently used in this industry right now. So that's a summary of what we're doing. I have no visibility of who is on board, so I just go on my next slide just to thank people at MIMS, and also to tell you that MIMS is growing fast. We're looking for a lot of people right now. We are growing at the pace of 2 new employees per month. So please, if you're interested to hear more and you're passionate about machine learning and AI and really interested and already have experience in life science, particularly, just do not hesitate and contact me at sarah@mims.ai or directly going to our website. And I'll stop the presentation so that maybe I can see somebody and take any question you might have. So thanks a lot for your attention. Do not hesitate to contact us. Please apply for internship or whatever. We're actively looking for people. So, and we have a very great team. Very happy to introduce the team to you as well. So thanks for the invitation. And I think I stop here if you don't have any other questions. Thank you. Bye.